In exercise 6, we'll be looking at the process of cell division, and this is a crucial process because this is one of the primary features that is used to distinguish life from other things that are out there, because living organisms can reproduce themselves. So there's a lot of vocabulary associated with this, and remember the difference between mitosis and meiosis, and we'll go over some of these terms here. We'll look at a little bit of the cell cycle and take a look at what's going on with the movement of chromosomes during eukaryotic cell division with mitosis and meiosis, and see how they produce differences in terms of the chromosomes, and those will give us differences in terms of diversity of organisms that are out there. So we're not going to be focusing on the prokaryotic version of cell division. This is going to be looking at eukaryotic cell division. And when you look at eukaryotic cells, we can really divide up the cell cycle into two different phases. One of them is what's known as interphase, and you can see the interphase itself is broken up into three different parts here. You've got growth phase that occurs at the beginning of this interphase cycle, and this is where the cell is doing metabolism. It's making new polymers inside of the cell and basically getting a little bit bigger. And then we distinguish the S phase, and this is where the cell is doing the synthesis of DNA molecules. So they are actually making a copy to get ready for cell division. And then the final part is this growth phase, where the cell is again going to get all the stuff together that it's going to need, because once they're done with interphase, they can enter into what's known as the M phase. And M phase technically could be mitosis or meiosis. And when you look at this, look at the big amount of time that they spend in interphase. This is estimated to be about 90% of the time of a cell's life it's going to spend in interphase, and then 10% of that time in this mitotic phase, where it's going through some characteristic events in mitosis or meiosis. And at the end of that process, you have cytokinesis, which again literally means cell moving, and the product of this M phase is you're going to get your cells that need to move apart there. And when we look at some of these structures here, we're going to be simulating chromosomes. And a chromosome is a interesting kind of structure, because normally when a cell is in interphase, it's going to be reading parts of its DNA. And so the DNA has to be unwound for it to get access to the instructions that are built into that chromosome. And those instructions are found in the genes. And we can see this idea here. A gene locus is just referring to a specific part of that string of chromosome with the instructions that determine the characteristics of the cell for that particular gene that is there. When we look at this process, you run into a little bit of fun with the terms, because a normal cell in that G1 phase has this single strand of the chromosome. When that is copied during the synthesis phase, now you have these two sister chromatids, and this is referred to as a chromosome. So it's a little bit weird that a single one by itself can be referred to as a chromosome, but also you have these two sister chromatids. And what we're looking at here is when the cell is getting ready to divide, they're going to take this long strand of DNA that is partly unwound as they're reading the instructions, and they're going to wrap up this string of DNA to condense it. And I always use the analogy of, if you're using a ball of yarn, you have to unwind it in order to use it. But if you want to move that ball of yarn from one room to another, you wind it up. You don't drag it around with the yarn hanging off and you're tripping over it. So cells are going to do the same thing. They're going to condense this DNA as they go into the M phase there. And so what we're looking at here with these two sister chromatids making up this chromosome structure is your condensed DNA right there. And then they are attached in what's known as the centromere here. And this is actually where your cytoskeletal elements are going to be attaching to the chromosomes so that they can move it around. So thus we have kinetochore. Again, the kinet right there. The motion. When you look at kinetic energy, it's the energy of motion. So this is the attachment site where your cytoskeletal elements are going to attach to this big chromosome structure so they can move it around. So obviously we're not going to be moving around balls of yarn. Instead, we'll be moving around chromosomes that are made up of beads. And then the centromere in the middle here actually has some little magnets in it. So we can stick these two things together, but we can also separate them. So you'll need to pay attention to the length of the chromosome, and we're going to have two versions that I'll show you in a second when we look at the video and go through this demonstration of the movement of chromosomes that are made from these little bead structures, or what's known as pop beads, so that we can move them around and also disassemble and do what's known as a recombination, where we move a chunk of DNA from one chromosome arm to a different chromosome arm. When we're looking at this process with our pop beads, make sure that you're looking at these sorts of things here, because I'm going to be asking you to make drawings of what you see with our pop beads at these different phases in mitosis, and to also make sure that you use two different colors when you're doing this process, because we're going to have chromosomes in a diploid organism, which means they have two copies, one from mom and one from dad. And so we do want to be able to distinguish between the different versions of the chromosomes that are out there. And notice at the very end of this process of mitosis, there's two circles, because you have two daughter cells that are coming out of that process there. The other thing is we have a variety of questions that you'll be able to answer in Canvas. And notice it's asking about chromosomes, which color and length. So that's why I want to make sure that you pay attention. I'm not concerned about you drawing the beads. It helps to draw circles for the beads so you know which one you're looking at in terms of the 3 or the 5 beads, to give you some distinguishing characteristics there.
So here is our simulated chromosomes that we're going to use to demonstrate the movement of the chromosomes in the cells during both mitosis and meiosis. And the reason these are different colors is so that you can assume that one set of these chromosomes comes from one parent and the other set comes from the parent of the opposite sex. So when we're looking at a diploid organism like a mammal like us, you're going to find that you got one copy, say, from dad and one copy from mom here. And in our simulated cell, we're just going to say that they've got two different chromosomes here, one long one and one short one. And so if we look at your chromosome in a normal cell that's out there, they're going to have a single string like this, and then you have your centromere in the middle. And these are kind of cool because they have a little magnet inside, so we can actually sort of demonstrate the duplicate with the chromosome so that you have your two sister chromatids. So if we look at this, this is how the cell would look during interphase. The one thing that we can't show is during interphase, part of the chromosome is going to be unraveled as the DNA is actually being read. And then during the process of cell division, you can distinguish that because that's where that long string of DNA is going to get tightly wound up so that it can be moved back and forth inside the cell. So we'll start off with our cells like this during interphase, and then the DNA gets duplicated, so we'll bring in our sister chromatids and hook them up in that centromere. So now we've got our DNA that has been duplicated. So when you start out and you're making your drawings in the lab manual, usually what I recommend is for the interphase, you can just draw these circles of DNA all over the place there because it really hasn't condensed until it goes into the cell division phase there when they're going to undergo mitosis and meiosis, like I said. So now we can look at this is these guys would be floating around inside the nucleus normally, and one of the signs that the cell is going into the cell division phase is that you get that condensation of your chromosomes. So not only do you get the condensation of the chromosomes, but you also start to see that nuclear envelope. Remember that double phospholipid bilayer. It's like the cell membrane, but there's actually two layers around there. That starts to dissolve. and breakdown, and then your DNA with these two sister chromatids that are forming this chromosome, those are floating around inside the cytoplasm for the cell now. And at the beginning of this process, these are just sort of in these random locations. And then during that first part of mitosis, when you go from prophase into metaphase, that's where that spindle apparatus is formed in late prophase, and it's hooking up to these little guys in the middle here, and that's where the microtubules are going to hook up, and they're going to line these things up in the middle of the cell. And so here we can see that we've got these things starting to line up, and I'll put them all along the midline of the cell. And so if we were to draw this, you would see the spindle forming, and you would have one part of it over here, and then the other part of it on this opposite side here, and they would extend to the midline of our sister chromatids. And now we've got our chromosomes in metaphase, and you can always remember meta, they're along the middle there. And then they're going to go into anaphase. And at anaphase, you're basically going to be breaking these apart, and your chromosomes are going to start moving to the opposite poles of the cell, like this. So this would be anaphase. They've broken those homologous chromosomes apart here, or I should say the sister chromatids have been broken apart, and they get dragged to opposite sides of the cell. And so I'll finish off this process, and this is telophase, where you actually are going to have this movement as these things move out to opposite sides of the cell. And then you're going to have your cell membrane forming in between these two as the cytoskeleton pinches off the membrane, and it's going to drag it in, and you're going to get what's known as a cleavage furrow. And then the cytoskeleton is going to continue pinching off the membrane, and that will cause these two things to separate. And the nuclear membrane or nuclear envelope is going to form around these chromosomes. And when you look at both of our cells here, think about what did they start out with, and what do they end up with at the end of this process of mitosis? We should have two cells that are genetically identical as a result of going through the process of mitosis.
This and put it on the microscope, and here you can see to a slightly higher power, bring it into focus, and here you can see the fish blastodisc at 100 times or 400 times total magnification and with mitosis. But if we scan around, you can see this, and we provide this sometimes for students, but the staining is not as distinct as what you will see when we look at the onion slice, because the plant tissue tends to hold together a little bit better because of the cell walls. Whereas if you've ever had a fish egg, you know they're pretty squishy. And so I just wanted people to be able to see that you can do this process in animal cells, but it is complicated just by virtue of the structure of the cells there. And when you scan around again, the majority of cells that you're seeing are in interphase. They are not going through cell division. And you'll be able to see that sort of diffuse staining of the unwound chromosomes in the actively growing cells. And here again, you can see some more cells that look like they're going through mitosis with telophase and also anaphase there. So we'll take a look at our onion cells that are undergoing mitosis, and as you go through the video, you can just pause as needed when you see those examples, and then I'm asking people to sketch some of those cells in a variety of circles like this, and then submit your drawings on Canvas. So we'll be looking at our onion root tip, and you can see on the bottom of this slide, that's where the tip of the root is that's growing fast, and I'm just scanning through so you can see all the different tissues, and the dark spots are actually the nuclei in the cells have been stained so the DNA is visible. We switch up to 100 times total magnification here, and you can see they're magnified a little bit more, and you get sort of a better image as the cells are magnified. But we still need to go up to 400 to really see the architecture of the cells. So I want you to scan through and look for different examples. Think about the beads that we looked at with the arrangement of the chromosomes during mitosis, and make your sketches of different cells, focusing on those chromosomes that you can see in the nuclei. And remember, interphase cells have diffuse gray staining, and you can see the nucleolus where the ribosomes are being assembled there. So in this part, we'll take a look at meiosis using our pop beads. And similar to the last one, go ahead and as you watch the video, make some drawings of the beads as the chromosomes are being manipulated. And remember to try and use two colors. And notice also again, at the end of the first phase of meiosis, we do end up with two cells that you can draw there. And then also we have a variety of questions to answer pretty quickly for these things after the completion of meiosis 1.
All right, so here's our chromosomes again. We're back to our interphase cells. And here we've got our two short chromosomes and our two long chromosomes again. And this would be during interphase. Again, we can't really show that heterochromatin where the DNA has been unwound because it just doesn't work for our purposes. So we'll go ahead and show that the chromosomes have been duplicated. And we have our sister chromatids are stuck together here. And now we're going to illustrate the process of meiosis. And remember, when you're looking at meiosis, this is where the cells are going through this reductive cell division. They're going to reduce the number of chromosomes that are in the cells. So at the beginning of this process here, same kind of thing happens that we saw in mitosis. The chromosomes are going to condense. You're going to start to see that nuclear envelope breaking down. But one of the biggest differences is these homologous chromosomes. So say long chromosome from dad and long chromosome from mom, these are going to line up. And this is where you can actually see that exchange occurring between your strands of cells. And so I'll see if I can show you this little thing here. We can take this and break off the arms from one, and this is literally happening inside the cells there, and you're going to form a new strand of DNA. We are literally recombining the DNA and mutating it on purpose. And so here we can see this particular set of homologous chromosomes. We're just going to show there was one strand break, and then the yellow arm of the chromosome and the red arm of the chromosome were exchanged between these two. So when we look at it, these middle chromatids right here are actually recombined. These ones on the outside are still the same sequence, but these ones have changed. And if this sequence is in the middle of a gene that's being read, that could potentially change the characteristics of that gene. So when we're going through meiosis, we're going to make cells that are genetically different from the parent cells. That's one of the differences. And so we can also line up our homologous chromosomes for the short ones. We're just not going to do a recombination event for that. So now these are going to line up in the middle of the cell. Again, that midline, when we're looking at metaphase, and this is the first division when we're looking at meiosis. And during the anaphase part of this cell division, these homologous chromosomes are going to move to opposite sides of our cell. And so now we're going to end up with two cells. This is one set of chromosomes for the cell, and this is the other set. And at the end of telophase, again, these are going to finish moving to opposite sides of the cells there, and your nuclear envelope can form around these things if it's needed to. But during meiosis, they're going to go into that second cell division here. And when you look at this, think about what we're looking at here. Is this cell diploid or is it haploid? Well, look at this. This has got two sister chromatids for the short one and these two sister chromatids for the long one. So they've changed their ploidy. We've got a duplicated chromosome. We don't have one from mom and one from dad here. We've got these sister chromatids that both came from mom in this cell, in this example, and our sister chromatids that came from dad, along with this recombined chunk over there.
In this part, we'll take a look at the second part of meiosis and finish off this reductive cell division using our pop beads to demonstrate what's going on with the chromosomes again. And for this, notice there's two circles because we ended up with two cells as a result of the first division in meiosis. And then the second one is going to end up with us having four cells at the end. So there's going to be two sets of circles to be drawn for prophase 2, metaphase, and anaphase, and then a total of four of them when we do get to telophase there. And again, like the previous one, there's a few quick questions to answer to consider when we go through this process about some of what's going on with the second phase of meiosis and the resulting cells that are produced as a result of that.
So when we look at this, what's going to happen? You're going to go through that really short prophase here because the cells don't really need a long prophase. They're not going to go through that recombination event in the second one. And remember, when you're looking at something like eggs, women started out this process when they were still inside their moms, when they were going through fetal development. The cells that were destined to become their egg cells went into prophase 1 and they stopped in prophase 1. And they waited until once a month The hormones were released that caused those cells to finish off this process of meiosis. So literally, women have been sitting there with their eggs ready to go, or actually not ready to go, partly ready to go, since before they were even born. Now, when we look at finishing off this process with the second phases of meiosis here, we're going to go through a similar thing. You're going to have them line up in the middle for metaphase 2. So there we have these things lined up in the middle. And then you're going to go into anaphase 2, and that's where these sister chromatids are going to break apart in both of these daughter cells that are coming out of this process here. And so these are going to get hooked up to the spindle apparatus. One of them would be here, and the other one over here on this side for this cell. And then this cell is going to have one of them here and one of them here. And they're going to start dragging these chromosomes out by hooking up to that kinetochore that's in the middle right there with your spindle apparatus attached there with your microtubule proteins. Remember those cytoskeletal elements that we saw way back in the earlier chapter where we looked at the parts of the cells. In late telophase, these chromosomes, or I should say sister chromatids, have separated and they've moved out to the outside. And so here we can see there's 4 different cells that came out of this process. And look at the number of chromosome strands that we're looking at in the cells. These are no longer diploid because we don't have the yellow and the red in this cell. We just have the long and the short red. We don't have the one that came from the opposite parent. This cell ended up with the recombined strand right there. This cell got the other half of that red one, and then this one here has the short and the long chromosome that came out of that process. So at the end of meiosis, we have 4 cells coming out of here. And if you look at and think about the chromosomes that are in these cells, they are no longer diploid cells.
We'll end off this looking at Mendel's law of independent assortment. And what this is dealing with when we're looking at the independent assortment is this idea of our homologous chromosomes. And again, when you're looking at this idea, you get 1 chromosome from one and 1 chromosome from mom and 1 from dad. And when we look at humans, we have 23 pairs of chromosomes. So you have chromosome 1 from mom and chromosome 2 from dad. And in this picture, we can see the duplicated chromosomes. So here, you've taken this one side and made a copy of it so that you are ready to split these apart so that each of the daughter cells can get a copy of it. But when we're looking at homologous chromosomes in meiosis, remember it's reductive cell division. So we're going to be separating this strand from that strand, but also we're going to be separating mom's sister chromatids from the ones that came from dad here. And so when you go through this process, how these line up makes a difference. And when we look at humans, remember, there's 23 pairs, which means the first chromosomes, you could have, say, mom on top and dad on the bottom side of the cell. The second chromosome could be the same thing, or it could be flipped. So when we look at this probability, there is 2 possible combinations for 23 different positions for human chromosomes. And so that is 2 times 2 times 2, and you do that 23 times, and you're looking at millions of possible combinations that can come out of this process of independent assortment. That can give us genetic diversity. And so when we look at this process, take a look at your chromosome drawings from anaphase 1 that you made earlier. There's a 50-50 chance that the beads are segregated differently. They could be either all red on one end and all yellow at the other, or if you flipped them, the short and the long chromosomes, you could have a mixture of the red and yellow ones at each end of the dividing cell. And if we take a look at the video there, at the end of anaphase 1, here's our cell with the chromosomes moved to opposite sides right here. You can see we ended up with all red chromosomes at one end and all the yellow at the other end. And when we went through anaphase 2, we separated each of those sister chromatids at the end of this process. So this cell is going to end up with a long and a short red. This cell is also going to end up with a long and a short red. But if you think about that process, what if one of these things was switched, so that instead of having both red here and both yellow there, they were switched. So now you have a mixture of those red and yellows. And so we're going to go through that process, and I'll show you taking these beads, and we'll go through that, and we want to take a look at the end result here. So we want to look at the end of telophase 2 by using this different arrangement of chromosomes that are coming out of that process from the first phase. So how they line up during metaphase 1 makes a difference in how they end up at the end of this process.
So at the end of our first cell division, we ended up with this pattern. And if you look at this, the red ones were grouped on this side and the yellow ones were grouped on this side. And I'm going to back up and go back to metaphase 1, where these guys were lined up. And the way they lined up, all the red ones ended up on one side and the yellow ones ended up on the other side. Well, what happens if during that first metaphase, where these short ones are lined up here and the long ones are lined up over here, what if these are switched? This is another possible arrangement. Notice the difference. If these guys are going to this side and these are going to that side, you're going to start to end up with a different combination of these things at the end. So we can take this and run it through the process of cell division, and we'll take these and put them over there. We'll end up these here. And now when we look at the 4 cells that come out of this, notice the previous one we had both sets of reds, but now we've got a red and a yellow. So if you think about this, this daughter cell got the short chromosome, say, from dad, and the long chromosome from mom. And likewise for this one. When we look at these guys, we've got a mixture of these different chromosomes. Rather than all the red being in one cell, we've got a mixture. And so when you think about this process, remember, you got your chromosomes from your parents. Where did they get their chromosomes from? Their parents. So this can change the types of genes that are on the chromosomes that you got from one parent versus the other. And here the 4 daughter cells are a complete mixture, with the traits coming from mom and from dad, depending on the chromosome that we're looking at. In our first round, each cell ended up either with the all red or the all yellow chromosomes. And so when we look at this process, this is giving us a different pool of genes in these 4 cells compared to the 4 cells that came out of the previous one, even though they all started out with the same components. They all had the two long chromosomes and the 2 short ones. The difference was the red versus yellow. So think about the difference that we get as a result to how these chromosomes, the homologous chromosomes, line up during metaphase 1 can influence the pattern of these chromosomes that we get coming out of the final process of meiosis.
So we'll finish off exercise 6 and just do some summary questions on cell division. And when we look at this process, remember, we can group organisms together based on their similarities. So animal cells share certain characteristics, but even when we go down to the level of cytokinesis during cell division, we can find some distinct differences between the process in animal cells versus plant cells. So see if you can figure that one out. You can take a look in your textbook and also in the lab manual to get some additional help if you need it. In terms of haploid and diploid cells, remember, haploid cells have a single copy of the chromosome. Diploid cells have duplicated copies, so they have those homologous chromosomes. When you're looking at this, remember, a zygote is a fertilized egg cell, and an ovum is a single egg cell that you're looking at there. And then this last question on this page is, how is asexual reproduction different from sexual reproduction? So remember, asexual reproduction is mitosis, and sexual reproduction is meiosis. You can also look at this process when you're considering a multicellular organism like an animal that is out there. We're doing sexual reproduction for the whole body, but within us, we're going to have asexual reproduction occurring as cells are dividing. And then at the very end are the toughest questions. One is to list 4 differences in the process and outcome for mitosis and meiosis. So when you're doing differences, it's a good idea to make sure you list both sides of that. So mitosis has this, meiosis has this, and find those different characteristic features that distinguish the 2 processes here. The last one is probably the most difficult one for a lot of students that are out there, because we want to find something that is going to give us an increase in variability of a species. Why are we using this process of sexual reproduction? It's giving us offspring that are genetically different from the parents. Well, there are 3 different mechanisms that give us genetic variability, and 2 of them are relatively easy to find if you read through the lab and you understand it. And there's a 3rd one that I highly recommend taking a look at the lecture video that I made, because that will help to point you in the right direction to be able to answer this.